Hey everybody, this is a video lab that's going to cover the pre-lab discussion for lab number 11, colorimetry. You're first going to start out this lab. You need to make a series of solutions using a process of dilution. The pre-lab questions will take you through how to calculate the concentrations and how to prepare those dilutions. You're going to do the calculations first, which will help speed up this process as you go. But basically, you're going to make a stock solution in the first test tube, and then you're going to take some of that and put it into the second test tube and dilute it down with water. Then you're going to take some of that original stock solution, put it into tube 3 and dilute that down with water. And then you're going to take some more out of tube 1 and put it into tube 4 and dilute that down with water, tube 5 and tube 6 and so on and so forth as it's laid out in the pre-lab. So make sure you follow those directions carefully. You should have already done the calculation, so it shouldn't take much effort, just simply measure and go. You might recognize these two pictures from a lab you did back in honors chemistry last year. And if this is your first go around with colorimetry, well then, welcome aboard. The picture on the left is what is known as a cuvette. And this is a small plastic cube that has two different types of textures on the sides. On one side, you'll see that's ridged texture, and on the other side, perpendicular to that, you'll see a clear texture. So you've got ridges on the front and back and clear on the sides. The cuvette is where you're going to place your liquids for analysis. Now when handling the cuvette, once you've got your sample inside, it's important that you only handle it with the ridged sides. The sides with ridges have texture, which is why you should touch them, and the reason they have texture is so it doesn't slip out of your fingers. Now sometimes these are made of glass, which means they're much more dangerous to handle. If you drop them, there's shards of glass everywhere. But these are all plastic, but you should still always handle it with your fingers touching the textured side, leaving the clear side for analysis. Now this apparatus over here is your colorimeter. It is going to be connected to this little device known as the LabQuest. And the LabQuest is simply a device that collects data for you. And so you can connect it via the probes on this side. There's channel 1, 2, and 3. It doesn't matter which side you plug it into, but as soon as you plug it in, the LabQuest is going to recognize the colorimeter, and it's going to pop up a screen that's going to be useful for data collection here in the next couple minutes. So this is a picture of the colorimeter from the top view, and I've zoomed in a little bit so you can see the different aspects. Some things you want to know is you first got the lid, which is open so that you can see the inner workings of the colorimeter. Inside is where you're going to place your cuvette. See this square? It's perfectly designed to fit one of those cuvettes. They're standardized, those cuvettes, the size, so that they fit inside this perfect square. These are a couple of different LED lights that are going to light up, and they're labeled in different nanometers, and we'll talk more about how to use that in a little bit. And then of course this button right here is called the cal button, or the calibration button. And you got a left arrow and a right arrow. Now the left arrow and right arrow is what you use to toggle between the different wavelengths of light that you want to shoot through your sample. These different wavelengths of light correspond to different colors of light, and those are going to shoot through your sample, and you'll be able to read some information from that, but I'll talk about that here in a second. The cal button is used to calibrate or zero out your colorimeter. You'd use this only when you have something inside that you want to calibrate. So you'd put the cuvette in with just distilled water, close the door, and hit cal, and that basically zeroes out your colorimeter. You don't want to hit the cal button when you have a sample of solution in there that you're trying to analyze, because it's going to zero out the entire colorimeter. It's going to basically recalibrate based on that color of liquid. You always want to calibrate once at the beginning and use only distilled water, since it's going to be the solvent of the solution that you're going to be using in this lab. Now how a colorimeter works is it analyzes a sample inside the cuvette, and what happens is there's going to be a light, and the light is inside the black box, but the light is going to shine, and it's going to shine light in this direction through the sample, and it's going to be picked up on a sensor on the other side. Now as the light passes through, it's going to lose some intensity because it's going to be absorbed by the colored solution, and the sensor is going to be basically telling you how much light has lost. The device knows the intensity of the light as it leaves the light source, and it also can tell you the intensity of the light as it hits the sensor on the other side, and it's going to be able to tell you how much light was lost to the solution, which is a measurement known as absorbance. So your absorbance value tells you how much of the light is absorbed by the liquid being analyzed. Now, the darker the color, the more that gets absorbed. The lighter the color, the less it gets absorbed. The data you're going to collect in this lab is absorbance data of the different samples from the different test tubes that you set up from the previous slide that I've shown you. Now, one more thing before I finish is, do you see this arrow right up here at the top of the colorimeter? The arrow is pointing back towards the middle or the inner workings of the colorimeter. This is the direction or the flow of light. And so when you set up your cuvette, you want the ridges to be on the sides and you want the clear sides to be facing away and towards the light, so that when the light passes through, it goes through unobstructed. We don't want it to pass through the textured sides because that would interfere with the light. We want the light to pass through only the cuvette and the liquid and not through the textured sides. So this arrow is a reminder, like the light's traveling this direction in vertically, so to speak, not horizontally. Last couple things I want to talk about here is a couple of notes here at the end of this presentation. Things that I see students do that would help speed up time. First of all, make sure you close the door of the colorimeter while you're taking the readings. I forgot to mention that in the video, but you don't want any light coming in and interfering with your absorbance data. Second of all, make sure you fill the cuvette only about 3/4 away to avoid spilling and damaging the colorimeter. Some students fill it up to the very top and then some spills over when they're handling the cuvette. Could damage the machinery. Next one is the lab quest should only be used to read the absorbance. You're not really touching anything on the lab quest itself. You're simply just reading the numbers and transferring the numbers to your lab report. Step number 4 here, or tip number 4, you don't need to zero out the colorimeter between every trial. It saves yourself some time. Just do it one time at the beginning and then proceed with your 5 different solutions. Make sure do it once at the beginning, that's all you need. Unless you get some strange numbers, then you might need to zero it out again somewhere in between. But always use the same cuvette every time for the zeroing process and for each of the trials. You don't want to have multiple cuvettes because not all cuvettes are made identical. And finally, number 5, use the supplied paper wipes to wipe down the sides of the cuvette because fingerprints could distort your absorbance data. I think that's all for this video lesson. Thanks for listening.